In this video, we're going to examine the origin of cells. Where do they come from? How do they develop? And what is the evidence and our understanding that supports what we know today about living cells? So the Earth is about 4.5 billion years old, and the conditions on Earth have changed greatly since its development, particularly the atmosphere. One major aspect that's changed quite a bit. Initially, during Earth's formation and the early years of its existence, really not a lot of oxygen on the planet. Trace amounts of oxygen, primarily because it reacted with other elements, for example, iron oxide. And so there really wasn't a lot of oxygen early on in Earth's history in the atmosphere or on the planets available for life, although life wasn't in existence yet. Not a lot of oxygen available. Methane was at a much higher concentration. Today we do have methane on the planet, particularly also greenhouse gas in our atmosphere, but in much greater concentrations early in Earth's history, primarily due to volcanic activity, as well as meteorites striking the Earth. And then also similar, carbon dioxide was much higher also in the atmosphere in Earth's early conditions because of volcanoes. Because of those things also then somewhat going hand in hand, temperature also varied quite a bit. Typically it would be higher than what it is today, primarily partially due to a lack of an established atmosphere, but really variable because the sun itself was not emitting as much energy, about 20% less energy, and so that varied the temperature. Comets, asteroids, meteorites also hitting the planets changed the temperature. And with different atmospheric components or no atmosphere, the temperature varied quite a bit. And overall, although we're not exactly sure of the specific range of temperatures, difficult to determine that far in the past, it's pretty accepted that the temperatures were overall higher on the planet. And also with no atmosphere, really had a lot more UV radiation. The atmosphere helps to block a lot of the UV radiation that the sun releases and comes to Earth, and without an atmosphere, a lot of that wasn't blocked. And so more UV radiation on the planet. Because of all of these things, the reactions that were able to take place on the planets were quite a bit different than what they are today. Vastly different, really, than what they are today. And formation of carbon compounds would be able to happen because of the different atmospheric components and the gases that were on the planet. And so carbon compounds probably formed in particular areas like hot springs or deep sea ocean vents that are very high temperatures. Places like those probably allowed carbon compounds to be able to form. And eventually these carbon compounds really could be in collection called a soup, like a soup of all of these carbon compounds. A collection of all of these carbon compounds could have been the building blocks of life. For example, like carboxylic acids, aldehydes, amino acids, and bases, of what would then go on to potentially make DNA and RNA. All of these could have developed because of the conditions on Earth early in its history. Again, those conditions don't exist today. Our environmental conditions, temperature, UV, atmosphere are very different than what they were in the past. But these earlier conditions could have allowed these carbon compounds to be able to form. And so that's potentially is how some of these first initial building blocks of life would have developed. Once life did begin to evolve, particularly with photosynthetic bacteria, it rapidly changed the planet, and particularly our atmosphere, with developing an ozone layer, a higher proportion of oxygen available on the planet, about 20% oxygen, and far less carbon dioxide. And so the Earth's early prebiotic conditions and atmosphere were very different than what they are today. Cells represent the smallest unit of life, and a single individual cell can carry out all of the functions of living things, and all living organisms carry out the functions for life. And those would be, for example, responding to stimuli, growth and development, use of energy, homeostasis, reproduction. Those would be different functions of living things. And regardless of whether the organism is a single cell or a multicellular organism, those living things are able to carry out all of the different functions of life. And living things can be composed of one or more cells. Typically, single-celled organisms are going to be prokaryotes. They can also sometimes be eukaryotes. There are single-celled eukaryotic organisms, but typically prokaryotes. And what makes cells the smallest unit, smallest unit of life is their ability to be self-sustaining. They can reproduce, they can pass on genetic material, use energy to maintain an ordered state, they can be cultured. All of these characteristics provide evidence that indicates that cells are the smallest unit of life. Although cells have smaller organelles or aspects inside of them, those individual pieces are not able to be self-sustaining. And while some organelles do have some of the characteristics of living things and may have evolved to become part of cells, they're not able to carry out all of the different functions that a living organism would be. And so really what this tells us is that cells are the smallest piece of life, or the smallest unit of life, and life is built from that, from single-celled organisms to multicellular organisms that are composed of millions of different types of cells. Today on Earth, we know that cells must come from existing cells. And if you think about the cells that are inside of your body or any other living organism that reproduces sexually, where did that initial cell come from? Your parents. Sperm and egg combine to form a cell that then becomes a zygote, and over a series, many times a series of divisions, that single one cell then divides and divides and divides to become the offspring that contains millions of different cells. And so today on Earth, cells must come from pre-existing cells. And we know this through a series of experiments. Louis Pasteur is a scientist, French scientist, that first helped to demonstrate this and provide some evidence from this. You probably have heard of Pasteur or pasteurization, particularly with dairy products. In Louis Pasteur's experiments, he took a series of glass beakers. They're typically called swan neck beakers, kind of like an Erlenmeyer flask, but with a swan neck, as you can see in the picture, and put some broth in each of these different beakers and then boiled them to kill off any bacteria or anything else that was living inside of them. And in one of the containers, just left it as it was. In another, after boiling, broke off the swan neck so that the broth itself was available to be exposed to particles in the air. And after leaving for a period of time, found that bacteria was growing in the beaker that contained, that had the neck broken off, and the beaker that did not have the neck broken off had no life. And so this helped to demonstrate that in order for life to develop and to arise, it has to come from previously existing life. And so Pasteur's experiments and our understanding of where offspring come from, whether it's asexual or sexual reproduction, today on Earth, life must come from pre-existing life. But then that begs the question: where did the initial cells and living things come from? And if life doesn't spontaneously now appear on Earth, how then did life evolve on Earth? And that's a big question, and one that scientists have some answers to and some hypotheses, but is still one that we're trying to understand and to culture more evidence for. And so if cells were to have evolved from non-living matter, it would really require a series of intermediate steps over a long period of time, and would require a couple of key developments to happen. One, there'd have to be some sort of catalyst to be able to take control of the reactions taking place. We previously just talked about the very different conditions on Earth millions of years ago. There would need to be some ability to be able to control what reactions were taking place. Second, there would need to be an assembly or self-assembly of carbon compounds into polymers. For example, the formation of amino acids into proteins, the ability to combine carbon compounds into larger molecules. Third, there would need to be some sort of compartmentalization, the ability to have an internal environment separated from an external environment. So there would need to be some sort of membrane that would need to be established or developed. And lastly, there would need to be the opportunity for self-replication of molecules, particularly for variants that were most successful, so that the successful variants could pass on to a future generation, like what we see in cells now and organisms now today, the ability to pass on for inheritance. And so all of those different hypotheses and those conditions would need to be present in order for life to develop from non-living life. And again, these are the understandings that we have currently based off of the evidence, but this is still really unknown, and we're still trying to discover and answer some of these questions of how cells could spontaneously develop. So we know that these carbon compounds are essential for the formation of life. How then do we know that these carbon compounds could have developed? And that was answered back in the late 1920s. 1929, a scientist by the name of Haldane suggested that maybe the early atmosphere on Earth's early atmosphere could have produced organic compounds. Energy would be for that would have been provided by lightning or UV radiation. He really thought that that oceans were a solution of organic molecules, and he called it a primitive soup or a hot dilute soup of those carbon compounds, and it contained all of the necessary components to make those carbon compounds. And he based that off of the presence of water vapor, carbon dioxide, and ammonia being in the atmosphere. And so one other possible potential source of carbon compounds may also have been comets delivering organic compounds to Earth. We've also found more recently that different asteroids contain some organic compounds. And so under heavy bombardments of Earth about 4 million years ago of different asteroids, they could have maybe delivered those organic compounds and water to Earth. So two different possibilities. And a little bit later on, more recent experiments have tested Haldane's hypothesis to see could the early Earth's conditions actually produce organic compounds. So in the 1950s, two scientists, Miller and Urey, actually set out to test Haldane's hypothesis and his proposal, and their experiments using a series of different glass beakers and containers set out to test Earth's early atmospheric conditions and simulated the conditions that what we think were present on early Earth and tested for the occurrence of chemical origins of life, organic compounds. And so adding some different gases, water, methane, ammonia, and hydrogen, and sealed them inside of this sterile container. After a couple of days, the water within it began to turn pink, and then after a week it turned red. And what were those colors? They were indication of carbon compounds actually being developed, including about 20 different amino acids that had been produced inside of this sealed, sterile environment just by adding those different molecules, water, methane, ammonia, hydrogen, and providing a bit of energy. And so using sparks to stimulate lightning, within a couple of weeks, about 10 to 50% of carbon within the system was in the form of organic compounds, with about 2% forming amino acids. And so this experiment of Miller and Urey in the 50s helped to show that it is possible, the abiotic synthesis of organic molecules is possible under conditions that we assume to have existed in early Earth, and provide some evidence that the abiotic synthesis of organic molecules is possible and could have been the building blocks of living things. And so two different hypotheses: the abiotic synthesis of organic molecules, or potentially the delivering of organic molecules through comets and asteroids. So having established the development of carbon compounds, the next crucial step would be the formation of some sort of membrane or vesicle, a capsule to be able to separate the internal and the external environment. And cells today are composed of phospholipids. And phospholipids we'll talk a little bit more about in the future, but they are composed of two main parts, a phosphate head and some fatty acid tails. And the phosphate head is hydrophilic. It will interact with water or liquid, whereas the fatty acid tails are hydrophobic. They tend to not like water. They don't mix with water. And surprisingly, if you take a number of phospholipids and put them into water, they will self-arrange so that the phosphate heads are exposed to the liquid and the tails are protected or not exposed to water. And they'll self-arrange and do this themselves. They're obviously just atoms and elements put together, but they will arrange themselves in this formation. And due to the polarity of the membrane, the hydrophobic heads, the hydrophobic tails and the hydrophilic heads, polar molecules would not be able to cross that membrane. They wouldn't be able to move through those hydrophobic fatty acid tails, and this could lead to an internal environment that's different than the external environment. And while this is not the formation of a cell, this is those intermediate steps that would be needed to form cells. And it's very similar in creating an internal and an external environment. And so this is potentially how vesicles that have an enclosed membrane and separate internal and external environments could have developed. So after discussing the development of carbon compounds and the formation of some vesicles, a membrane, our last big piece to look at the evidence is the ability to pass information from one generation to the next. And what was this material that allowed this to happen? It was probably RNA. Life has to have the ability to transmit information from one generation to the next. There needs to be a genotype, that information. So for us today, that would be the sequence of DNA. Our A's, T's, G's, and C's are different nucleotides. And then that information needs to be expressed, and that would be referred to as the genotype. And RNA, very similar to DNA as we've discussed, RNA can store, transmit, and replicate genetic information. And so because of its ability to do all of these things, it probably was the first source or type of genetic material. As we'll talk about later in the course, enzymes, types of proteins, are essential to making proteins themselves and for living organisms to be able to survive. Enzymes are types of proteins, and they're essential for life. And so what evidence do we have that suggests RNA preceded DNA and maybe could have been this first source of genetic material? Well, one, viruses use RNA. They typically have a high mutation rate. Viruses have much less DNA than other living organisms, whether or not they're living. They have a very high mutation rate due to inaccurate replication process. The molecule, the enzyme that does the replication, is called polymerase, and it's not very accurate in viruses. And so in a changing environment, actually having mutations and creating genetic variation would be very beneficial. RNA stores information in the same way as DNA does, but it is self-replicating, and it can actually act as a catalyst itself. RNA can act as a catalyst. Some of the processes that are catalyzed by RNA, for example, would be ribosomes. A ribosome is what makes proteins. We'll talk about a little bit later on. And a ribosome is composed of RNA. And so it's used to make protein. So that's how it can be used as a catalyst. And additionally, experiments have demonstrated that different variants of RNA could undergo natural selection. Some could be more favorable than others. And as we'll talk about later on, natural selection being the driving force for evolution, we could see some different types of RNA having a better chance of survival, a higher degree of fitness in the particular environment, and so they could actually be favored. And so RNA is that first genetic component of living cells and living organisms, and could have provided that means to be able to pass information from one generation to the next. Today on Earth, we have three domains of living things: the bacteria, the archaea, and the eukaryotes. And all of those organisms, whether they're single-celled or multi-celled organisms, they all use genetic code. The sequence of DNA, A's, T's, G's, and C's, it's universal for all species. And what makes each of those species different is the amount of that code and the combination of that code. And within the genetic information, there are 64 different types of codons, and it's a sequence of 3 bases. And those 3 bases can be combined to form 64 different codons, and each codon is unique. We'll look at this a little bit more in the future, but each of those are unique in that they code for or have the instruction for the phenotype for a particular type of amino acid, alanine, for example, or they are start or stop codons. They start the process or stop the process of combining amino acids. And so small variations within that sequence can result in very different species. And organisms that have more similar code inherited it from an ancestor. So two species that are very similar are going to have inherited that from a common ancestor, because cells come from cells and cells contain DNA, and so each one of those cells has the DNA, and the similarity between them would indicate how closely related they are. It is possible for organisms to have really similar structures or DNA without a recent common ancestor, and this is particularly due to convergent evolution, where the environmental conditions, the selection pressures that organisms face, result in similar characteristics, whether they be internal or external, that result in some similar structures, and that's due to convergent evolution, where they don't become the same species, but they become more and more similar, like, for example, a bat wing and an insect wing. They have a similar structure of wings with the ability to fly because of similar selection pressures. The advantage of flying is best, and so we see those develop. And so how we know that or organisms are related is, for example, the ribosomes and the enzymes that are involved in the synthesis of DNA and RNA are nearly identical in all organisms. DNA and RNA is found and used in organisms to be able to survive. How those actually are synthesized are done so with ribosomes and enzymes, and they're nearly identical in all different species. Other different types of enzymes or proteins are also similar. Cytochrome C, Ribisco is very common, used in plants for photosynthesis, the Calvin cycle, and that sequence of DNA that's responsible for making that is similar in all organisms. So we can track and follow the genetic code, and our ability to synthesize, to sequence DNA now, the technology that exists to be able to do that. So we can really look at different species and make comparisons and follow the genetic code and see how it's changed from one species to the next, and even extinct species if we're able to acquire DNA from those extinct species. And this allows us to identify most recent common ancestor. And while there's not one particular organism that we've identified this, the most recent common ancestor when comparing different species, the going back all the way, we can find the last universal common ancestor, and this is referred to as LUCA, the last universal common ancestor for the species that exist on the planet today. What then is the evidence for this LUCA, the last universal common ancestor? How old is it? What do we know? The earliest uncontested evidence for life comes from some rocks in Western Australia. They're known as the Stirling Pool Formation, and they are large fossilized stromatolites. And these form from mats of cyanobacteria in shallow seawater that trap sediments, and they secrete calcium carbonate. And through dating, radiometric dating, they're dated to about 3.42 billion years ago. And so this is our uncontested best evidence for how old life is in this last universal common ancestor. There is some other potential sources of life that predate that or even older than that, but they're less concrete evidence. The earliest life would come from isotope ratios. Due to heat and pressure, they're metamorphs into rocks. So there's really no clear fossils that allow us to be able to assess this. And there are a couple other locations that have some of these metamorphs rocks, some in iron rock in Greenland, and it dates back to about 3.7 to 3.85 billion years ago. It may be the remain of stromatolites, I'm not entirely sure. And then also in Western Australia, the Jack Hill rocks are a little over 4 billion years ago and are consistent with the biogenetic origin of life, but not proof. So probably the best, most safe evidence is the stromatolites from the Stirling Pool in Western Australia, and indicates that last common ancestor is about 3.4 billion years old. All organisms on the planet have DNA, and gene sequences, DNA sequences that occur widely of different groups, indicates relationships and how closely related the species are. And bacteria and archaea, because they're in different domains and have been on the planet for much longer than eukaryotes, if bacteria and archaea have sequences that are similar and they match the evolutionary tree of modern organisms, this suggests that they were inherited from an early common ancestor. There's about 355 different protein families that were likely inherited from LUCA, primarily needed for anaerobic metabolism, carbon dioxide and nitrogen fixing, and this would suggest an environment with high concentrations of hydrogen, carbon dioxide, iron, all conditions that are found around hydrothermal vents in the oceans. So the hydrothermal vents in the ocean are essentially cracks in the ocean floor where there's gushing hot water that have lots of inorganic chemicals. They're typically extremely hot, 60 to 90 degrees Celsius, and they have high concentrations of hydrogen, methane, and sulfides. And these hydrothermal vents may have been the source of earliest cells, and particularly the carbon compounds that helped to make up those earliest cells. And so because of the presence of these different inorganic materials, the temperature, the pressure, these all are conditions that could have led to the synthesis of these organic compounds that then could have been used to form the earliest pieces of life.